Hey everybody, let me start with an apology. I really had intended on being there live, and the vagaries of life got in the way, and too long to get there, back, day jobs, all those kinds of things. And so I'd also like to tell you that our plan B is for me to Skype in at the end of this talk. You're looking obviously at a recording, and I will be live and answer any questions that you have. So I'd like to thank Jerry for the invitation. I'd also like to thank Jerry for his patience and understanding of me not showing up there and doing this thing live. So here we go. I've been looking into anomalous healing now for, I don't know, 125 years, something like that. And by anomalous, I mean in a classic Jungian sense, stuff that shouldn't be. So healing that is not normal or doesn't fit into the textbooks, things along those lines. And the kind of healing that I've looked into primarily has sometimes been called healing with intent. Sometimes it's called hands-on healing. It's along those lines. And the hands-on healing that I've looked into doesn't have anything to do with the kind of things that you might have heard of. So, for example, Reiki or Jo-Ray or things like that. I don't do comparative healing research. I've simply devised a non-believing healing technique that seems to produce pretty interesting results. And so my research agenda has been, and I hope that I have a pointer here, to investigate the parameters of anomalous healing. And by that, I mean, among other things, what makes it work well, what makes it work not so well. Does it matter, for example, whether the healer believes or the healee believes, or does a state of mind matter? And I'm not going to talk about that today, but it has some interesting areas that are maybe down the road we'll talk about. But for example, I think that all other things being equal, which is of course a famous statistical cop-out, but all other things being equal, belief seems to retard healing, which I find reasonably interesting. The second bullet, I'm interested in the physiological correlates of healing. When healing happens, or when you try to heal, what happens to the healer, what happens to the healee. I have a whole variety of data on functional MRIs and EEGs and EKGs and a bunch of other Gs. And so that's an interesting area if anybody wants to talk to me on the side. I'm interested in whether healing can be taught, and can you take an inexperienced skeptic and turn them into a healer? I'm not a healer myself. The fourth bullet point, development of a theoretical model. I got a long ways to go. I got a long ways to go before I have anything reasonably approaching a theoretical model of how healing happens. It happens, but what's the mechanism? It's reasonably speculative to me at this point. And so, taking almost a flip side approach, right now I'm interested, and the purpose of today is the development of practical applications. Even if we don't understand the thing, can we capture it? And can we reproduce it? Can we reproduce healing without a healer? Can we turn this into a conventional therapy even? I think is a reasonably interesting question. So this last one that's highlighted will be, I think, of particular significance to this conference. You folks are the water people, and you know much more about water than I do. And so the question is, you obviously know the Benveniste stuff, does water have memory? Can water remember healing and reproduce the healing? Can materials remember healing and reproduce the healing? Does cotton work? Does water work? Do a variety of other materials? So we'll look into that a little bit. So these kinds of questions are allowing the theoretical part, which would be wonderful, to kind of take a back seat and just say, can we reproduce it, even if we don't fully understand the mechanism? And it turns out it's a pretty interesting question that has a lot of methodological complications, which I will spend a little bit of time talking about today. So I've done a variety. I'm going to focus on my cancer experiments, and I've done cancer experiments in vivo as well as in vitro. This page is entirely in vivo, so we're dealing with mice experiments. I'm hoping my pointer is working there. This mammary cancer, I've got City University of New York, and over here, I hope, I have 2 experiments, that's what it stands for. So I've done 2 formal runs of the mammary cancer at City University, 2 at St. Joseph's, 1 at ASU, 3 at Indiana University Med School, and 1 is currently running at Brown University in Providence. University of Connecticut Medical School on a sarcoma. And I'm going to focus in the in vivo stuff today on these experiments, or a selection of these experiments. But I also have experiments on oncogenic mice at UC San Diego, nude mice. That would be my racy talk. I don't know you well enough to go about on nude mice. But at Brown University, we have some of nude mice, extremely aggressive cancers. And I will finally today talk about this last one, an in vitro question of human leukemia cells that was also done at University of California in San Diego. So the emphasis will be on these in vivo experiments, some of them, and the in vitro experiments on human leukemia cells. And it turns out there's some reasonably interesting data. The mammary cancer that you saw, a bunch of these. I'm on my 14th mouse experiment, and more than half of them were done on a mammary cancer. This is biological gobbledygook. That the thing that really matters is this last line, which says 14 to 27 days. And the real short version, any biologist knows what's going on here, or certainly an oncologist. You take cancer cells, certain kind, put them into a certain kind of mouse, you inject them with a certain number of cells, and you follow them after that. And this particular mammary cancer is, I'm speculating here, but it's probably the most widely studied cancer model of all time. The bottom line is the bottom line. Host survival, 14 to 27 days. And what this really means is you inject the mice with cancer, and 100% of them die 14 to 27 days subsequent to injection. It forms almost a perfect normal curve. The standard deviation of death is 3 days. This is a very, very tight model. And there are literally thousands of published experiments on these. So I use I do unorthodox things to these orthodox models. I'm not inventing some exotic thing to study. I'm using standard lab stuff, and I'm doing healing stuff to it, and we find out what happens. That's really the plot line. This particular model is very easy to study because it's non-metastatic, and the tumor simply grows and grows and grows, and the mouse dies an ugly death, either from malnutrition and or having the internal organs crushed. It's not a good way to go. So I'm going to show you some ugly pictures first. And this is what's supposed to happen. So you got a mouse, and the tumor grows, and the tumor really grows, and I hope the pointer's working, but this is the tumor. It's essentially swallowed a leg. You can see that the skin is not good, the fur is falling out. If you could see the eyes of this mouse, it's not really there. It's not a good way to go. And so this happens 100% of the time, and again, it follows a nice, almost perfect normal curve distribution, and you do stuff to these mice, and you see if you can change the statistical distribution. Can you get it to be negatively skewed? Can you extend the life? Can you? But before me, the world record was 27 days. And what we do to these off-the-shelf laboratory mice is we do this healing technique that I've developed. The healing technique has nothing to do with belief. I actually show you a picture of me. This is a picture of me. I tend not to use pictures of other people, but this is a picture of me at Indiana University. I was not the only person doing these healings at Indiana, but I don't want to show other people get them. They don't want to be famous. So I'm sitting there holding a cage of mice. I'm doing my mental technique. The mental technique, if it looks meditative, it's misleading, because I assure you I'm not now meditating. In fact, I'm doing the technique right now. But I'm running through very rapid imaging, I'm holding the cage. I'm actually wondering how my life got to this point. I'm sitting in a lab holding a cage of mice. And how did I what road led me to this crazy place? But I'm sitting there and I'm doing cage after cage. There are other mice rooms going on at the time. And in other rooms, they're doing cage after cage, and we do a whole lot of mice in these experiments. So we treat, and by treating, I've also done dose experiments. Does the number of treatments matter? The length of treatment? You know, all these kinds of things that we don't have time to really look at today. So I thought, to give you an idea how little I know about this, I thought that if healing were to work, and this is years ago, and I got to them early enough, so I'm thinking of it analogously to radiation, something like that. So I get the mice and I heal them, and I somehow intend something to happen, that nature would bend to my will, and healing would happen. Right? If I get mice soon enough after they've been injected, and I go zzz, this zapping that I would do would prevent the tumor from growing. Something along those lines. And this was in my arrogant period. And so I was thinking of healing as something analogous to a PK effect. I'm intending a particular outcome, and I can intend the means to get to the outcome, and that turns out to be the height of arrogance. I can intend an outcome, but I certainly don't predict what happens. So instead of going zzz, zzz, and the mice never get cancer, the cancer grew. In fact, in the first experiments, I was really convinced that the thing was failing, because I was trying to keep the mice from developing tumors. The tumors developed, the tumors got bigger, I'm going zzz, and nothing's happening, and then the tumor starts to slit open, and now I'm sure they're dying. And then the slit turns into an ulceration, and it turns into an ulceration, and now I'm really sure that they're dying. So I'm intending, in thinking this is something analogous to a psychokinetic effect, I'm intending that something specific happen. Nature's not going the way I intended to go. I tried to call off the experiment a whole bunch of times, just assuming it was failing. A heartless friend of mine talked me into keep going. And so you're going to see a couple of slides now of what happens, and it's reasonably interesting. So again, here's a mouse. This is the number of days subsequent to injection. And you're looking at a tumor that's growing. I presume that this wasn't working well, and the tumor starts to slit open, and the tumor starts to ulcerate. Now, this doesn't look like a happy mouse, but look at the fur compared to the previous mouse I showed you. This fur is looking good. If you could see the eyes are sharp and clear, and this dopey mouse is running around in the cage having a ball. It's running around, it's playing with its cage mates. Remember the world record is 27 days. This is 35 days subsequent to injection. And if you follow this, and I'm not going to follow it on this mouse, but if you follow this, the mouse tumor implodes, and the mouse is cured. And by cured, I mean cured for its life. I used to say it remits, but biologists started yelling at me and said, this isn't a remission, it's a cure. And you'll see full cures in a little bit. This is another example of slightly less aggressive cancer. This is a sarcoma. And if you look at this line here, I've got dosage. We're not kidding around when we do this. We at least double the lethal dosage. I mean, we're really trying to kill these suckers, and we can't seem to do that very well. But in this particular case, this is another nice model to work with because it's not metastatic. The thing just grows and grows, the same kind of thing. And we do the treatments, and by we, again, it's me, it's other faculty members, it's students, it's people crazy enough to be in my experiments. And you'll see what happens if we do the treatments. In these particular mice, the tumor goes away in two different ways. Again, think it's not conforming to my will, it's doing whatever it does. And in this particular mouse, you come in one day and suddenly it's like somebody let the air out of the tumor. And of course, you know, because I can't help it, the mouse mysteriously grows an arrow on its back. But you can see that the thing just completely was eliminated. Somebody let the air out. Or some of the mice, and we have no idea why this is, some of the mice develop an analogous ulceration pattern and then implosion of the tumor. It's pretty interesting. And then once the thing is gone, there's no sign that the mouse ever had this. And again, these things will live the entire lifespan. So again, it's an interesting phenomenon. It's not just one particular kind of cancer. It's a bunch of different kinds of cancer. Cancer gets cured. We're trying to figure out why, and we're trying to figure out if we can reproduce the effect. And I mean, I have other data too. So I have humans. I just did this for a time sequence. So February 10th, March 21st, you're looking at some things, obviously breast cancer. On the left, you don't want to see, and like 6 weeks later, you're looking at things you do want to see. Humans don't like it because they don't like, you know, to fit to those little cages. But we can do this through thermal imaging. These are week-long exposures. You start out looking at things you don't want to see, and the things that you don't want to see eventually go away. So we can look at thermal imaging, MRIs, blood counts, tumor counts, all these other kinds of things. And so it's not limited to mice, I mean, pretty honestly. And an interesting observation about healing is that it seems to occur in bursts. So you don't have, here's a condition, after a treatment, after another treatment, after another treatment. There's no linear relationship at all. It seems closer to a phase transition. So all of you water folks, you take water and you cool it a little bit, and you cool it a little bit, and cool it a little bit, and suddenly it changes and it turns to crystalline patterns. And so in your group, of course, you've got 4 phases of water, and so how do the transitions happen? I'd like to know that. But it seems to be sudden, and sudden and dramatic. Like you saw earlier, the mouse, as if somebody let the air out of the tumor. A tumor suddenly imploding. We're not sure exactly how to think about this, whether it's cumulative dose of healing, or whether there's a phase transition, or whether it's unknown. But I'm going to show you a couple of really nice pictures because they're worth watching. Here's a mouse, 22 days subsequent to injection, and you see an ulceration, and you see how clear the eyes are and how good the coat is and all this. I'm going to show you the exact same mouse 6 days later, and this is a biologist pulling away the fur, which has already grown back, looking for where it was. And again, I'll do it again because this is very cool. So 22 days after, 28 days, all across, in this particular experiment, there were 6 cages of mice treated by 6 different people, and everything just suddenly changed in those 6 days. And so we're not sure how to think about that. And then if you follow this mouse 35 days after, there's no sign this thing ever had cancer, and it will live its entire lifespan and be fine. So the real short version to the phenomenon is this. After you do this healing, and I don't like energy healing because I don't think there's an energy. We've never had a mouse that had a recurrence of cancer, so they are cured for life. And among the interesting things in highlight here are the mice are immune to repeated injections of cancer. And this is for their entire life. And an infected mouse can be cured by cell transplantation from a treated mouse. Also pretty interesting. And once you have done a treatment to a mouse, the cell line will no longer seed forward. So you get cancerous mice, you grow a tumor, you take the tumor, break it up, you inject cells into the next generation, and it seeds forward, and it seeds forward, and it seeds forward. Once we do the healing, the cell line will no longer seed forward. Reasonably interesting. Now, speculation, and I'm not sure if I'm asking the right questions. Maybe there's an immune factor that's going on here. Maybe there's a memory in the cells somehow. Maybe there's a memory in the water. Maybe there's a, maybe. And this is, frankly, what I'd love to hear your suggestions. And if anybody would like to help unravel how cancer cures happen, I'm very, very open to ideas as to how this thing works. Because, boy, I'll tell you, this is, it's largely speculative on my part. So, putting this into more show and tell, on the left, we have a treated, ulcerated mouse, you know, it's ugly looking. And we don't obviously have one mouse, we got a bunch of mice. And on the right, we have a treated mouse, an untreated mouse, has never been subjected to healing with tumors. And so we take some cells from the treated mouse, and we re-inject those cells into the untreated mouse that's never been exposed to healing, and everybody gets cured. Reasonably interesting. And I'm going to ask a slightly different question, and this is, I think, the most directed at you folks today. What if healing is directly applied to materials? So I'm going to talk about treated cell medium, and I'm going to talk about treated water. And treated means doing healing. So one of my experiments, actually I've done it a couple times, but healing cell medium. Now this is straight off the shelf medium for in vitro work. You gotta grow cells in a medium. They need nutrients and all that kind of stuff. Here is an off the shelf standard lab cell medium. And what we do is we take the bottles themselves, and we put our hands around the bottles. So we're actually treating a cell medium. Now, the cell medium presumably has no need of healing, and we're putting our hands around and we're doing my healing technique, but you know there really is no need for healing. And so this is pretty interesting. So the cell medium is treated, and it's given to a lab. And then the lab independent, no healers walk in, independently, they grow and they play around with the cell cultures with this treated medium. So here's something from UC San Diego. In the first week, nothing anomalous happened. So we've got cell medium that has not been treated, cell medium that's been treated, and then we grow the cells. This is human leukemia cells. Nothing happens, nothing crazy is going on. The second week, though, and think of sunbursts that we just talked about. The second week, suddenly, the medium treated leukemia explodes. It goes wild. And by wild, I mean we're averaging a little bit more than a 600% increase in cell growth. Now, a 600% cell rate increase in cell growth, we don't need to do statistics on that. So the 600% increase, so the cancer in the cell, in the well plates, just goes crazy. Now this may be another way to think about healing. Instead of thinking about healing as psychokinetic, think about healing as giving the client what the client needs. And if a mouse has cancer, and it's got a tumor here, the mouse needs to get rid of the tumor. If you have human leukemia in a well plate, what does cancer like to do? Well, it likes to grow. What happens if you offer it healing? Well, cells in a well plate want to reproduce. Nothing happens the first week, and then the second week it explodes. I think it ate 2 or 3 techs in the lab. But it explodes in growth. And so something obviously very anomalous happens. One of the things, I haven't done this yet, I got a whole bunch of experiments on the drawing board. One of the things that I'd like to do is to take this supercharged cancer and put it into a mouse And my guess is it'll collapse. It's a guess. But I suspect that healing goes to the higher order need of the organs. We got to touch this. We'll find out, hopefully, down the road. We also can treat water bottles. So this is just your basic water. And the way you treat a cell medium, and the way you treat mouse cages, and you feed that to mice you never meet, and those mice also get cured. That's interesting. So we can cure, brief summary, an infected mouse by cell transplantation from a treated mouse. We stop the ability of the line to seed forward. We can treat a cell medium, which apparently will affect the growth rate. It doesn't, I mean, if you're not a fan of cancer, it's going in the opposite way you like, but it's having a tremendous effect on the cancer. And mice drinking treated water apparently will be cured. And so, at least on the surface, it seems like it's very encouraging. But there's a phenomenon of resonant bonding, and this makes methodological problems, to say the least. In our healing experiments, the experimental and control groups can become, I mean, I have on the board there entangled. I don't like the word entangled, I like resonant bonded. If you give a treatment to an experimental group, it seems as if we're also giving treatments to controls. Now, one of our experimental faith-based assumptions, and I think it is faith-based, is you take a group of whatever, mice, cells, people, and you split them up into two groups randomly, and you treat one group and you have another as a control. The faith-based assumption is that physically separate groups are independent. What if that's not right? What if it's possible that they can bond together? What if they're not completely independent? And you can see where all sorts of type 2 errors can result from this. I actually have a series of experiments I'm about to start where I suspect that placebos, in fact, are really instances of resonant bonding, and we're interpreting it wrong and thinking there's something psychological going on. I suspect that it's more physical than it is psychological. So it's a pretty interesting area. I don't have time to do this in a whole lot right now, but you can get the general idea here. So here's another experiment. Down here, this is pretty enlightening. Down here, I just have treated cages. So think these bottom cages, and there's more than 3, but think that there's more, that these are cages treated by people using my healing technique. And at least 100 meters away, we have controls, and obviously the healers never see the controls. And we also have, I didn't know about this until the experiment was over, a second control group not known to the healers. Didn't know. So people knew there would be control cages, more than one. People didn't know there would be a second control not known to the healers. Now, this is the white mice that you saw before. This is the sarcoma model. And these are obviously randomly taken from the same batch of mice having been given cancer. But over here, these labs and these labs are working on the exact same model. So they're working on the sarcoma, and they're doing conventional biological research. These are getting healing. These are not getting healing, but they're from the same batch. But these are the white mice. Now, here's another question. How should we think about healing? If it's a field effect, and I don't know if Rupert Sheldrake is among you, but Rupert, if it's a field effect, Rupert's idea would be that there would be a morphogenic field, which, if these were fixed and put out a field that might fix this, then these in between ought to similarly, since they're the same mice, similarly be affected. But here's what happened. The treated cages got cured, the controls not seen by the healers got cured, and the second controls not even known by any of the healers to even exist got cured, and None of the intermediary labs were affected at all. Same mouse model. How do we think about this? It's a pretty interesting problem. I don't think field effect is the answer. And it's still not so simple. Resonant bonds appear to be fluid. You can make them, you can break them. And speculatively, I mean, everybody knows what a resonant bond, everybody knows what it means to make a connection. So you can be, you can connect to your dog. You can love your dog on Monday and hate your dog on Tuesday. You can love your car on Monday and hate your car on Tuesday. And so it's not the car, it's not the dog. The bonds are fluid. I'm attached to the dog, I'm not attached to the dog. You can be attached to people, places, things, ideas. And sometimes I'm not attached. I think the question is, what are the rules of bonding and what are the rules of unbonding? And I think this will go a long way in terms of trying to figure out how placebos work. So I want to give you a very, very, very interesting experiment. This is with the mammary cancer. We have treated cages. We have a control lab. This is in the same building. And we have a second control off-site. Now, these 3 cages, and there really were 3 cages, were treated by biology students. They didn't believe in healing. They actually thought I was doing a study in gullibility. They didn't believe in healing. They did my technique. They did all this. And so their task in this experiment was to treat these cages an hour a day. There were 2 cages treated by non-biology students. The biology students were told there was a control lab in the building someplace, but don't go looking for them. And that, of course, means I'm going to go look for them. And so the biology students said, okay, we won't look for them, but we'll just go peek. And they went peeking, and then they came running back, and they didn't tell anyone they peeked. Non-biology students didn't know what a control lab was. And here's what happened. In the lab, all the biology students' mice died. Biology students can't cure in a lab. Non-biology students were able to cure. The control mice that had only been visited by the biology students were cured, and they only saw them for a couple of minutes. And the second control site that was shipped to another city, different lab, they died on schedule. Now, we also did, in this particular experiment, we did something that IACUCs, I don't want to say this, but I also gave everybody cages to take home. Don't tell any IACUC people. So they, not only did they treat in the lab, but they also took cages home. And they took them independent, so nobody saw anybody else's mice. And here, everybody got cured. All the mice got cured. Biologists can cure at home. Biologists can't cure in the lab. This actually goes in, this is in, similar to what the great Bernard Grad at Yale University, the father of healing research, found. He found out that medical students, biology students, actually retard healing. And so I'll just leave that to sit there. But the biology students tense in the lab, their mice die. Biology students relaxing at home can cure their mice. I'm thinking of this as bonding, increasing and decreasing the strength of bonds. More to follow. So some questions. If there is resonant bonding and physical separateness doesn't mean independence, was it actually the cell transplants that did the curing? Was it actually the medium that did the curing? Was it the treated water that did the curing? And you can see that this is a pretty difficult methodological problem. If bonds are fluid and non-local, how do we find the source of the cure? And that's what I'm looking for help in trying to understand that. It's a tough problem. So, among the research questions is, can cotton and water alone produce cures with no other healing going on? Can cell transplantation work with no other healing going on? Can we detect some sort of a signal? So We're looking at this stuff storing material. Is it a biological storage? Is it a physical storage? Is there a signal? Is there a difference between treated and untreated cotton and water? And if so, can a signal reproduce the cure? These, I think, are reasonably interesting questions. In other words, can we take this crazy anomalous healing and can we then turn it into a self-replicating cure? Whether that cure comes from biological memory, immunologically, or otherwise, cell signaling, I'm not sure. Could it be, is there something in the water? Is there something in the sun? And then can we transform it into a conventional therapy and take all the craziness out of this? I think that's something that probably is worth looking into, and I welcome your ideas, thoughts, and questions. So I'll leave you with a Adobe cartoon that somebody made up for me, and I hope everybody there can read it, but just for fun. And thank you for your attention, and if all goes well, I will soon be joining you with Skype. So hopefully I'll see you all soon.
Hi, Bill.
Hey.
Hi, it's Jerry.
Hey, Jerry.
Hi. Nice to see you here in Bulgaria as opposed to Seattle. Question, you never mentioned the rate of success in your initial slides in curing the mice. What percentage of the mice were cured?
If the volunteer healers got the mice, I mean, we changed the parameters of healing so many times that it's not a simple answer. We got different denominators, but if the healers got to the mice within about 3 days after they'd been injected and they had sufficient time to treat them, the cure rate is about 95%. There's a couple of mice that I can't account for, but for the most part they get cured. Among the experiments we've done is dose response questions. So we've tried reducing the amount of treatment and such, and there seems to be a minimum amount of treatment necessary to produce the cure. I just don't know exactly where the line is.
Bill, this is Glenn.
Hey, Glenn.
Hi. So I have a question about your tissue culture experiments, and
okay.
I wanted, since you were successful when you treated the water, but not with the tissue culture medium, and one of the things that we learned in this conference is that different waters with different mineral content receive information better than others. And since tissue culture medium has proteins and DNA and all kinds of chemicals in it, I wonder whether those chemicals might be absorbing the information and keeping it to themselves. So I guess I'm suggesting that the next time you do an in vitro experiment, treat the water and then make the culture medium from the water. Any thoughts?
It's an interesting idea. First of all, let me disagree with your premise. I think we were wildly successful with the treated medium. It just wasn't in the direction that you might expect if you were doing a PK experiment. That is, if you're looking at success as cancer being killed, then yes, it was a very strong failure because the cancer cells were wildly stimulated to grow at a very anomalous rate. So at least 600% increase in cell growth. So something is very much happening, and my interpretation of this is not that it failed, but rather that that the healee
in this particular case, cancer cells, takes what it wants from the healing, and that conceptually, we ought not to think of healing as me imposing a particular outcome on nature, but rather nature taking what it seeks. So a cancer cell in a dish wants to replicate, and if it's treated, or if it's grown in treated medium, then it replicates wildly. I think that if you're dealing with a mouse that has cancer, it wants to get rid of the cancer. And I suspect that if we took this wildly replicating cancer and injected it into a body, then it would have a very different outcome. So I don't think it failed. I think it showed a very strong response and leads you away from the idea that healing is, I want a particular outcome and my will or intention will get that outcome. I don't think that's the case at all. In terms of the types of water, yes, I think that this could be very important. And I've been told, for example, never use distilled water. And different types of mineral water and such, I think may be a very important variable. So I will also be in touch with you and Jerry and folks for input on that, and that's a very interesting observation.
Dr. Bengston, there is Vladimir Vayek from...
Switch it on.
Hmm?
You have to unmute it.
Yeah. No.
I'm not hearing.
Oh, do you hear me? That's Vladimir Vayek asking you a question. I'm deeply impressed with your presentation, and it reminded me about a lot of, there are a lot of parallels, it seems to me, with the observations of Wilhelm Koch, who was treating, curing cancer, thousands of cases in 1920s, 1930s in Detroit. He was using the highly diluted homeopathic remedy based on methyl glycol, glycosylated, and things like this. And if do you know about his works? Wilhelm Koch, medical doctor.
Yes, I do.
Yeah. What do you think? Was he healing with homeopathy or with himself?
I didn't catch the last question.
Did he treat with a real remedy? Or he was treating cancer, curing cancer by himself?
Are you asking what was the source of the cure?
Yeah. Was it Wilhelm Koch as a personality, or he...
I don't know, but the experimenter effect seems to be particularly strong in healing research. Just as you folks know that, for example, Benveniste water memory experiments seem to also have an embedded experimenter effect, and so I think that the key in this to find out whether it was a person or whether or not it's a reliable independent effect will come when we try these experiments to replicate it, and the farther we get away from anybody's personal attention on this, we'll start to get a glimmer in terms of whether it's a person, whether it's a resonant bond, whether it becomes something now in nature that can self-replicate a cure. I don't know the answer. I'm kind of hoping that it'll be a self-replicating cure, but Benveniste was also hoping that the memory was independent of the person in the lab. So nature's pretty quirky.
Thank you very much. And also I would like just to note that he observed the same thing as you, that cured people, they got strongest immunity, and they practically never got ill after curing.
Yeah. Yeah, and I don't know whether, as we look into this, whether we'll find that this is, the mechanism is biological, whether it's physical, whether it's neither. I just got some data yesterday, actually, that there seems to be a biological effect of something like treated cotton. So if we take, I didn't talk about this in today, but we have experiments going where we take just ordinary cotton, we treat the cotton, and that seems anecdotally to have effects in humans. And we just got some in vitro data yesterday that suggests that it also has biological activity that will be stimulated in cell cultures.
Some more questions? Okay. I think as it's... Oh, one more question, yes, please, here we go. Last one in this case, please.
Hi, it's Tracy Latz from North Carolina. I think I met you in a previous life. What were you doing with the cotton? Once you treated the cotton, did the people carry it around like their pet, or what did they do? I'm curious.
Could somebody repeat the question?
What did the people do with the cotton?
Okay, the cotton, you saw a picture of me holding hands around a cage.
Yes, I understand you said
essentially you're doing the same thing with cotton, and it's not a particularly mindful exercise. You take a piece of cotton, you put it in your hand, and this is also an interesting observation. If you move, it seems to make a difference.
But the people with the cotton, after you treated it, were they putting it in their ears, or what were they doing with it to carry it around and be healed?
The people doing the treating of the cotton would simply hold the cotton in their hands and use my healing technique, which is a very rapid imaging technique. The people who were receiving the cotton as patients or clients would just apply the cotton to a particular area and tape it if they could, and then the healing happens.
Thank you.
Thank you very much. Let's give another big applause. Thank you very much.